the pathogenesis of Staph aureus. So the genus Staphylococcus are gram-positive bacteria. There's lots of different species and subspecies. They produce staphylococcal exotoxins, lots of them, as I'll show you. And nearly all cases of staphylococcal infection are attributed to a specific species, Staphylococcus aureus, that's transmitted through skin and nasal secretions. It appears that both animals and humans are important reservoirs. So the main reservoir for aureus is probably people, but dogs and so forth can carry it as well. When we did a sampling of Cal Poly students, we found that about 20% were carriers of Staph aureus in their nose. And roughly about 2% of those, as I've indicated, were methicillin-resistant Staph aureus, which is a pretty, very deadly version of the bacterium that kills lots of people in hospitals, as you'll see. The microbes can cause true infections, as indicated here, or they can cause something called an intoxication, in which the microbe gets into food, grows, and produces a toxin. So even if you kill the microbes after it's produced the toxin, because the toxin is heat stable, the toxin can actually still make you sick. The slide here represents methicillin-resistant Staph aureus, and so it starts here with penicillin resistance, where the bacteria exchange genes for the production of this enzyme called beta-lactamase. And beta-lactamase specifically breaks this beta-lactam ring in the penicillin structure, so it produces this inactive product called penicilloic acid. So penicilloic acid can't bind to the bacterial transpeptidase enzyme that's necessary for helping build cell walls when the bacteria are dividing. And so this inactive compound no longer interferes with that transpeptidase protein, and so the microbes can continue to make their cell walls. So humans, being as smart as we are, came up with an end around to the beta-lactamase problem. So bacterium produces beta-lactamase, we produced methicillin. And the basic gist of it is that methicillin contains this dimethoxyphenyl ring that you see here. And this probably sterically, protects this cut site here. So beta-lactamase won't work. And therefore, even if the microbe produces a beta-lactamase, it won't work, and so the microbe can still be killed by methicillin. Just a bit of clinical data. It's a little old, but that's generally consistent. About 95,000 cases of MRSA in 2005 across the United States. About 19,000 deaths, and 2/3 of the infections occurred outside of the hospital, so they were community-acquired Staphylococcus aureus, or community-acquired MRSA, as we call it. 1/3 of the infections were nosocomial, or hospital-acquired. So, that's a lot. 6,000, anywhere from 6,000 to 10,000 people a year die from Staph aureus infections that they've acquired in the hospital. This slide is an overview of the virulence determinants of Staph aureus. And so you see here's our gram-positive coccus. Here's an electron micrograph, so you can see this organism up close. But this guy has a lot of ultrastructural as well as enzymatic armaments that allow it to be a really good pathogen. So first off, you can see that it's got adhesins that allows it to stick to host tissue. Another thing that it has structurally is this molecule called protein A on its surface that you see here that binds to antibodies, but it binds to them in the wrong way. So these are the antigen binding sites. So these antigen binding sites are what's supposed to be stuck to the microbe, but this kind of flips around so that the microbe binds to the antibody backwards. And so instead of having a neutralizing effect on the bacterium, it actually gives it a protective coating almost and shields it from an immune response. The microbe has a microcapsule, so it's a polysaccharide layer around here that allows the microbe to avoid or minimize being phagocytosed by macrophages and so forth. But then the other thing to notice is that it produces a whole bunch of toxins. Toxic shock toxin, for example, a superantigen type toxin that can overstimulate your immune response and cause severe reactions, kind of like that cytokine storm we keep hearing about with certain COVID patients. It also produces an exfoliation toxin, which damages your skin. And then these SEA through G, that's the emesis or vomiting toxins that I had alluded to. That these are the ones that cause the intoxications that can get into food and then get into your stomach and then cause a rapid vomiting response within an hour or two of eating them. So if you've ever gone to a party, for example, and where there was a food laid out for a while, and you get something that has Staph aureus in it, it's very possible that that you could develop symptoms very quickly. Again, because it's an intoxication, the microbe doesn't have to grow in your body, and therefore the incubation period is very short. Some of these other toxins that you see here are, the main ones are listed here. And so the microbe will produce like a staphylicin, for example, that you see here, which is this here. These are toxins, these are pore-forming toxins that act on red blood cells and can pop them open. The microbe also produces leukotoxins like leukocidin and leukotoxin. You can put them together in the leukotoxin category, but these guys act together to damage cell membranes of host cells. So if your immune response sends phagocytic cells and other things after these microbes, they can potentially kill them. The microbe also produces coagulase, which converts fibrinogen to fibrin to facilitate clot formation, which is a pretty important protective feature. So the microbe gets into a cut, immediately produces this coagulase to stimulate clotting, which allows the microbe to then grow in a protected clot. And then the microbe can produce an enzyme like staphylokinase that dissolves the clot so that it can spread, almost like a Trojan horse type effect. Here we see some of the diseases caused by staph, and they're quite variable. The soft tissue infections we're very used to. We've seen those, the skin infections and so forth are flesh-eating disease and things that we're often used to seeing. But some of the other stuff like menstrual toxic shock syndrome, which is typically caused by microbes that produce TSST in the urogenital tract, particularly of females, and this is often associated with improper tampon use. The microbes can also cause sepsis, meaning they just get into your bloodstream and cause your immune system to go haywire. And usually the combination of toxic products from the microbe and overreactive immune response can kill an individual. Osteomyelitis is one of the worst of it, where the microbe actually gets into the bone, and I've heard it's excruciatingly painful and very difficult to treat. Obviously, if the microbes are inside the marrow, the antibiotics that are prescribed are probably not going to easily get into those areas. Infective endocarditis is obviously very problematic when the microbes get into the muscles of the heart and so forth, and can actually damage the heart and cause heart attacks and heart failure and inflammation of heart valves and so forth. Lastly, but also pneumonia, where the microbes can get into your lungs and cause severe damage to the alveoli and inhibit your ability to breathe. So here's images of some of the toxins. Here's the result of EFT in two different scenarios. In one case, we see the scalded skin syndrome, where you have the microbes producing this toxin, and it's causing such an inflammation reaction that this little baby's skin is actually peeling off. And over here, the Staph aureus can cause something called impetigo, which is more of a superficial and localized infection of the skin. And so these are usually just diseases of poverty often, but they're also associated with close contact sports like wrestling, for example, where individuals are kind of rubbing up against each other. And so the lesions are typified by these brownish, crusty, they almost look like dried honey. As far as the lab is concerned, we identify the microbes through something called a coagulase test, or we can grow the microbes on blood agar, mannitol salt agar, and there's a variety of rapid tests as well. And I'll go through these guys. So the coagulase test is just looking for that enzyme that I showed you previously, coagulase, that causes the fibrin clots to form. And so we can do this in a test tube using rabbit plasma. you can purchase this stuff, and normally the plasma's is fluid, and you just take an isolated colony that you suspect to be Staph aureus and shake it off into the liquid, and then after about 2-3 hours of incubation, if the liquid clots, then you know that the microbe is Staph aureus. Another thing we can do is, as a throat swab or a skin swab or whatever, to blood auger, and specifically we'd be looking for growth, but also the production of staphylicin, which is evident by this beta hemolysis. So hopefully you remember that from the lab I just handed out, the lab handout I just sent around. And this clear zone around the bacteria on this sheep red blood cell auger is indicative of a staphylicin production and therefore beta hemolysis. Another approach would be to use mannitol salt auger, and this is what we gave you guys to, or would have given you guys to do skin swabs, looking for Staph aureus or similar staph on your skin. And so again, the medium looks like this, uninoculated, and so the microbes can be heavily inoculated onto the medium, and then you look for this characteristic reaction. So the medium is selective in that it has a high sodium chloride content. 7.5% is really high. I mean, you consider that's I don't know, twice as salty as seawater, at least. It has differential ingredients: mannitol as a fermentable sugar, and phenol red as a pH indicator. And so Staph aureus and other staph will grow on the medium, but only Staph aureus will vigorously ferment the mannitol to produce a yellow zone as a result of the acid production. And the last thing I'll mention here is the rapid testing. In this particular case, it's just a slide agglutination type test, looking for a specific penicillin binding protein in the bacterial cell envelope. And so this PBP2A is encoded by the MEK-A gene, and this particular penicillin binding protein is present in MRSA strains, or methicillin-resistant Staph aureus strains. So, as I talked about, the microbes become resistant to penicillin through the production of beta-lactamase, and so we produce in our labs we purify and produce methicillin to kill those microbes. Well, not surprising, the microbes become methicillin resistant then, or MRSA, by modifying this penicillin binding protein. In other words, they modify that transpeptidase enzyme that I mentioned that's essential for building cell walls. And so the modified penicillin binding protein no longer binds to methicillin, and therefore it can continue to work and continue to allow the microbes to grow. So this is an example of a rapid test that looks for that modified penicillin binding protein or transpeptidase. So in this particular case, you have these tiny little latex beads that have antibodies to the penicillin binding protein or transpeptidase on their surface. And then you apply the microbes and stir them up on one of these little plates. So imagine you put a couple drops of the antibody bound particle, and then you stir in some microbes from your pure culture samples, and then you look for clotting on these plates. And the clotting is because you get cross-reactivity, and these latex beads will clump together into a visible reaction called co-agglutination.